I would like to, so before I start, I'd like to show you, whenever I give a talk in public, I like to also share the slideshow. So this slideshow that you are about to see, it's going to be accessible on our website. The link is not active yet because I just sent the PDF to the webmaster, but it should be active very soon. And everything in blue in my slides will be a clickable link. So there will be references and everything. So if you download the PDF, you will have access to more data. So basically, what I'm going to talk about is about the functional definition for microRNA targets. You all know, or you certainly all know, that microRNAs are small regulatory RNAs which repress specific target RNAs which are recognized by base pairing. In fact, the best predictor that we have so far to predict microRNA targets is a perfect pairing between what people call the seed of a microRNA, the nucleotides 2 through 7, counted from the 5' end, and the target. Of course, there are exceptions, but this is the best predictor that we have so far. So when a target exhibits a perfect seed match to a microRNA, especially if this seed match is located in the 3' UTR of the target, then the target in general is repressed. And so that's how many computational programs could be developed for target prediction. They look for seed matches in 3' UTRs, and then they also select the ones which have been conserved in evolution, assuming that if the binding site has been conserved, certainly it must be useful to repress that target by that microRNA. So here, to make it super clear, I'm not talking about the same process that Sophie has just been talking about with TDMD, where the target was inducing a degradation of the microRNA. Here, I'm talking about the classical repression of targets by the microRNAs. And so if it's conserved in evolution, it is assumed to be functional and useful for the organism. It's not just about this. It's not just about looking for conserved seed matches, because in fact, many refinements can be added. And the latest version of Target Scan is, I would say, to my opinion, it's the best of these prediction programs available. In the latest version of Target Scan, which was described in this paper in 2015, they included many other context features which help predicting the affinity of a given target for the microRNA. This is an example of what a Target Scan prediction looks like. So this is in blue, that's the map of the 3' UTR of a gene, and all these small colored little things, these are predicted binding sites by this program Target Scan. For example, this binding site predicted for mu134 and so on. As you may see here on this particular example, there are many individual microRNAs apparently targeting the same mRNA. And that's not such a big surprise, because the seed is just 6 nucleotide long, so it's very short. And so even if you select the ones which have been conserved in evolution, well, you find many seed matches. And according to the latest estimations, more than 60% of the human coding genes seem to be targeted by microRNAs, which means that they have conserved seed matches in their 3' UTR. The problem is that because there are so many genes, then you find in the literature, you find a real zoo of all these papers, like saying that this microRNA targets that gene and controlling this and this phenotype. And the literature is now absolutely overwhelmed with papers with always titles like this, microRNA so-and-so is doing this and that by targeting whatever gene. So what I'm going to show you here is just 2 weeks of production by the international scientific community on this topic. I just picked 2 weeks randomly in the early 2015, I think end of 2014, beginning of 2014. In 2 weeks, that's all the articles that you can see on PubMed with titles like this: mir blah 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 is doing this by targeting blah blah blah. And so, of course, that's not a surprise, because so many genes are predicted to be targeted by microRNAs. So the main consensus coming out of all this is that microRNAs seem to be implicated in every physiological process in animals, because whatever physiological process you can look at, you can be sure that at least one of the genes in that process will be one of the 60% of target genes. But there are, and that's basically the consensus in the field these days, but there are several worrying observations which suggest that this may not be true. First one is that the effect of microRNAs on their targets quantitatively is extremely small. Usually it never exceeds twofold, and for most of the targets, they are repressed by like 10–20%. And that's not so much, because we all know that in molecular biology, even a twofold change is not so much. For example, we know that most genes are haplosufficient in animals, meaning that approximately twofold decrease in gene expression, gene transcription, doesn't yield any phenotype. And that's not even a twofold change in transcription, because normally after deletion of one, inactivation of one allele, already there are some feedback loops already modulating this fold change. So haplosufficient genes tell us that or more or less around a twofold change, twofold decrease in gene expression for most genes doesn't seem to yield any phenotypic defect. We could actually validate that more carefully on one particular example, a microRNA called miR223, which is specifically expressed in mouse neutrophils and the mouse neutrophil lineage. For that particular microRNA, so the effect of that microRNA on its targets was available already from this paper, actually from the Bartel lab. And so what we did is we actually measured the variability between wild-type individuals in the expression of each gene in their neutrophils. So from one wild-type mouse to the next wild-type mouse, how much does gene expression vary from one mouse to the next? And we saw that for typical predicted targets for that microRNA, the variability between wild-type mice was largely larger than the effect of the microRNA on these genes, which seems to imply that the microRNA doesn't do much on these genes, because already between individual wild-type mice, you see more difference. And also another worrying observation, this is the thing which is starting to pile up now that CRISPR made it much easier to actually investigate in vivo the real effects of individual microRNA target interactions. There are now more and more examples of cases where a given microRNA controlled phenotype had been really dissected, and so we can tell through what targets the microRNA could control this phenotype. And in each of these cases, each of the cases where it has been found, the phenotype seemed to be due to one single target, as if all the other targets which can be predicted computationally and which can be observed in molecular biology, all of these targets, they do really interact, but somehow doesn't seem to have any macroscopic consequence. And so that's really a paradox, because you may remember what I said earlier, that these computational programs, most of them, they work the same way. They look for seed matches in 3' UTRs and select the ones which have been conserved in evolution. And now I'm telling you that most of them do not even seem to be functional because they don't repress the targets enough. So then why have they been conserved in evolution? And so that's when we started to ask this question, that we started to consider that perhaps this very approach of looking for phylogenetically conserved seed matches somehow could also capture false positives. So let me explain a bit how it worked. This is an example. When you want to see whether a given microRNA binding site is conserved in evolution, for example, this miR134 binding site located in the 3' UTR of this USP9X gene, you want to see whether this binding site is conserved in vertebrates. So you can look at the autologous sequence of that binding site in various vertebrates. And in that particular example, that's the result. So the human microRNA sequence is written here. And so the seed is on the 5' end, here it's on the right. So the seed match is here on the right. And it's very well conserved among these vertebrates, except in mouse here with two mismatches. In the other ones, it's very well conserved. Also, you see a very good conservation of the block of nucleotides facing the 3' end of the microRNA, which is also something quite typical. So not surprisingly, this particular binding site is a very good bona fide target for miR134, according most of these computational prediction programs. But the problem is the miR134 microRNA itself is a very recent microRNA. It only exists in placental mammals. And here I'm not just talking about the miR134 microRNA, but about the miR134 family. What people call the microRNA family, that's all the microRNAs which share the same seed. So basically, there's no microRNA with the same seed in marsupials or in birds or in fish, and yet the binding site is conserved here, which means that the binding site is conserved for some microRNA independent reason. So we don't know what this reason is in that particular example. I don't know, maybe there's an RNA binding protein sitting here and it has to bind, and it's a very conserved protein, so that's why it has to be conserved between mammals and fish, for example. And just fortuitously, this protein binding site would overlap, would contain the sequence of a seed match to miR134. So that may be a reason. Maybe it's a DNA binding protein which has to bind there, we don't know. And so what's for sure here is that at least this binding site looks like it's been conserved because of its interaction with miR134, but in fact it's not. It's certainly conserved for some other reason. So of course this was just one particular example, but then we can look at how frequent that kind of thing is. And so for this we looked at various microRNAs in the human lineage because we needed enough data, and that's where we got most of the data. So what is shown here on the y-axis, that's the proportion of seed matches in 3' UTRs for any given microRNA family. So the proportion of seed matches which is over-conserved, which means that the seed match itself in the UTR is more conserved than the microRNA itself. And then we classified microRNAs by their conservation. So these are hominidase-specific microRNAs. And you see, for example, that for a typical hominidase-specific microRNA family, about 90% of its seed matches are more conserved than the microRNA itself, meaning that the seed match is conserved also outside hominidae. And you see very, very strikingly, the deeper the clade of specificity of the microRNA family, and the lower this false positive rate. To the point that when you look at utiliostomy-specific microRNAs, there's basically almost no overconserved seed match. One worrying possibility could be that, and that would be a bit expected, that perhaps hominidase-specific microRNAs, they are not so reliable. And we know that the least conserved microRNAs tend to be the least reliably annotated microRNAs. And perhaps these are the real microRNAs and they have no false positives, and this is more or less bullshit. So that would be one possibility. Another possibility would be that because we are looking at a tree, of course, you have more chances of observing an overconservation if you have more outer species to look at. So if you look at hominidae, there are many outer species. If you look at utiliostomy, there are much less. In fact, among the sequenced vertebrate genomes outside utiliostomy, there was just one, a lamprey genome. So of course, if you have just one outer genome, you have much less chance to observe overconservation. So that's what the other possibility. So that's what we decided to investigate. And what we saw is that regardless of the conservation of the microRNA family itself, well, we observe the same decrease when we look at overconservation in outer clades. For example, when you look at Catarini-specific microRNA families, well, we already see a decrease in the rate of overconserved seed matches from hominidae to Catarini, even though these microRNAs are present in all these clades. And we even see the same, in fact, that's the black boxes. We see the same when we look at non-seed hexamers, which are the hexamers which do not match the seed of any known vertebrate microRNAs. So this really looks like this decrease was mostly due to the fact that we are looking at a tree, and because of this this architecture as a tree, well, the deeper clades they have less outer species. So it looks like this effect that we measured for the least conserved microRNAs was certainly not artifactual. Another thing we had to check was that these computational programs I've been talking about, they don't just look for conserved seed matches in 3' material, they have additional criteria. I mentioned that very briefly a bit earlier. They look for local AU content and things like that. And so maybe they help cleaning up a bit the predictions. And indeed, they do a good job at reducing the false positive rate, because you see, for example, for hominidae-specific microRNAs, the false positive rate drops from 90% to about 50%. But still, depending on the programs, you still have large numbers of binding sites which are clearly too conserved to be true. They are even more conserved than the microRNA itself. In fact, some programs even probably they notice the problem, and so they don't even bother to predict targets for the least conserved microRNAs, or they predict the targets but without using conservation. That's the case for target scan, for example. To be absolutely fair, I have to also mention that so in our case, basically what we are proposing is that a large chunk of the predicted targets by these computational tools, they're just false positives. But some people before us have already attempted to measure this overconservation rate. And that's what has been published by a paper from the Bartel lab in 2009, where they basically used control k-mers, which are not matched by any vertebrate microRNA, to look at how much they are conserved. And so they were trying to use k-mers which have the same GC content, dinucleotide composition, and everything, which look a bit like the actual seed matches for microRNAs. And they were measuring how much these control k-mers were conserved, and then they subtracted that number to really see what was the excess conservation for actual microRNAs. But I don't think this is the best way to do, because of course by doing that they are looking at k-mers which are not the actual seed matches, which means that they do not bind, for example, they do not bind the same RNA binding proteins, and so they are not submitted to the same selective pressure than the real microRNA seed matches. So that's why I really wanted to mention that, because I have to say that we have a strong disagreement, Dave Bartel and myself, on this issue. I do not consider that this measure really measures really corrects for the microRNA independent conservation. All right, so and then what's the big deal? In fact, what about, I mean, okay, maybe there are many false positives in these predictions, but is that really important for biologists? Well, it is, because these computational programs are heavily used. And I'll just give you a brief example. The miR34 family. So be careful, don't mix. Before I was showing you the miR134 family, which was placental mammal specific. This is the miR34 family, completely different. miR34 is actually very conserved. This microRNA family is perceived as a tumor suppressor, according to a large number of studies. This is one of the most prominent reviews in the literature. Many people see miR34 as a tumor suppressor, it was believed that miR34 is frequently deleted. Actually, the miR34A member is frequently deleted in many human cancers. And it has been also believed that miR34 repress proliferation, cell proliferation. And so somehow, maybe miR34 would be a tumor suppressor by repressing proliferation of tumorigenic cells. We have this manuscript, Sophie, that you just heard. Sophie is the first author. So the manuscript is not published yet, but it's available on bioRxiv. So if you click this, you will see it. It's in review right now. We have good reasons to believe that this was mostly wrong. In fact, this microRNA family is not so much deleted in cancers, and the fact that miR34 is occasionally lost in cancers seems to be due to the fact that it's genomically close to a real tumor suppressor, which is CHD5. And also we have some good evidence that in fact it's not pro-proliferative as long as you're using endogenous levels of miR34. If you transfect crazy amounts of miR34, it's going to kill the cell, but that's not the actual function of the real concentration of the real microRNA in vivo. And one more argument why people believe that miR34, the miR34 family is a tumor suppressor is that when you look at the predicted targets for miR34, not surprisingly, you find a lot of them because, as you remember, the seed is very short. So for that particular case, the miR34 family, according to the latest version of TargetScan, you have more than 750 predicted targets. And of course, this is just the top of the list. But when you scroll down that list, of course, you will find genes whose names really could mean something in terms of cancer biology. For example, MDM4 or RAS. These genes could really be like pro-oncogenic or at least help tumorigenesis. And of course, if miR34 would repress these genes, probably miR34 could be anti-tumorigenic by repressing these oncogenic genes. This appears to be mostly wrong because in fact, the microRNA itself is not expressed enough in most of the tissues where it is believed to play an anti-tumorigenic role. And this kind of thing is completely neglected in the literature. And still now, like years after the in vivo effect of miR34 deletion was studied in mouse, still you can find many articles claiming that miR34 is anti-tumorigenic, and most of these papers would rely on the potential oncogenic activity of predicted targets for miR34. All right, so as a conclusion of everything I've been showing today, so you understood that we would like to revisit the definition for microRNA targets. For us, a microRNA target is not just an mRNA which is bound and repressed by a microRNA. The target, this repression has to yield some macroscopic phenotypes. And that's very important to consider that everything that you can observe in molecular biology, especially in high throughput molecular biology, which is so sensitive, everything you can observe does not always have a biological function. Everything you see in molecular biology does not always translate into an evolutionary selectable phenotype. For example, you can really see that whatever gene is repressed by 20% by whatever microRNA, this does not constitute a biological function. It has to affect fertility, viability, and everything to really constitute a biological function. And so the classical way to address that is to use phylogenetic conservation. By looking at conserved binding sites in evolution, you would say, okay, I have no idea why this particular interaction is important, but it looks like it's doing something important because it is conserved in evolution. In this case, we saw that, in fact, many binding sites for microRNAs, they are conserved for other reasons than the interaction with the microRNA. And this is not really surprising because when you think of the diversity of known microRNAs, like right now, there are about 1,500 annotated human microRNA families. And so when you group families by their hexamer, nucleotides 2 through 7, and there are 4,096 possible hexamers, which means that whenever you're considering, let's say, an RNA binding protein, and RNA binding proteins, they also have binding sites which are about like half a dozen nucleotides, the same kind of length than microRNA binding sites. So when you consider a given RNA binding protein, it's going to have a binding site which is more or less an hexamer. Well, about one hexamer out of 2 or 3 among the possible hexamers will be a seed match for a known human microRNA. So it's not a big surprise to see that, in fact, many of these RNA binding proteins, in fact, their binding sites would overlap with known seed matches for known microRNAs in humans, which means that when we come to this with a microRNA centric point of view, we would see this conserved binding site, and we may be tempted to assume that the binding site has been conserved because of its interaction with the microRNA. That's why in the case of microRNA binding sites, this like comparative genomics approach is finally far from being perfect. And so what I would suggest, at least when people predict microRNA targets and they use these computational tools, is first very obviously to exclude these binding sites which have been more conserved than the microRNA itself. And this is especially frequent when you look at very poorly conserved microRNA families. But also, this is something that we proposed in that paper that we published in 2017. We propose that the actual phenotypic robustness of a given gene can be measured by looking at the inter-individual variability among wild-type individuals. Genes whose activity is extremely dose-sensitive, these ones, they have certainly undergone a purifying selection so that they are under a very controlled expression, and so their expression is very stable from individual to individual, because any fluctuation in their expression would trigger a phenotype. Which means that if you look for those sensitive genes, which are the most likely real functional microRNA targets, you may want to look at these genes which vary very little from individual to individual. And also another thing which is very important to remind is that experimental validation is a real genetic knockout. Like you mutate the microRNA gene or the microRNA family genes, and then you observe phenotypes in vivo in a real in a real in vivo setting. When people do a reporter assay, they fuse their UTR of interest to a reporter gene, and then they overexpress tons of the microRNA, and they try to infer biological functions from this kind of experiment. This is extremely artificial, and this is not going to tell you that the microRNA really does repress that target. So this is a very dangerous experiment to do, and it's been very popular, unfortunately. And finally, I like to finish on this. As you may see, that what we are saying is that basically among all these microscopic events of interactions between microRNAs and mRNAs, most of them, they are real at the microscopic scale, but just their consequences are attenuated by homeostatic mechanisms, and they do not trigger any macroscopic phenotypes. And this is due to the fact that homeostasis is absolutely central and universal in the living world. In fact, I would define life as homeostatic mechanisms, things which remain more or less constant despite external insults. But of course, if homeostasis makes you robust to external insults, it also makes you robust to internal changes like gene regulation. In fact, there's a real antagonism between homeostasis and gene regulation. Gene regulation tends to modify the properties of the biological system. Homeostasis tends to attenuate these modifications. And because we know that homeostasis is so widespread in the living world, as we could see with haplosufficient genes, for example, it certainly means that, for example, negative feedback loops are much more common than positive feedback loops in biological systems, which implies that the extreme phenotypic sensitivity to small fluctuations in gene expression, which is required when people talk about fine-tuning gene expression, this may be true for just a few genes, but most of the genes, they are probably not, their biological activity is probably not sensitive enough to fine-tuning to really benefit from such a functional regulation. In other words, it looks like the butterfly effect, where a small effect on gene expression will have tremendous effects on the macroscopic phenotype. This has probably been counter-selected by evolution. The implication of that being that most of these events that we observe in microscopic biology are probably not relevant in macroscopic biology. All right, so I would like to thank the people who have been involved in that work, especially Sophie that you just heard. She's the main author on that study that we have right now on miR34. And I would like to thank, of course, all the agencies which supported our work financially. And I would be happy to take questions.